Okay. There might be a few more people who join, but I think we should probably kick off. If you're ready to go, Holly, I'll just do a quick introduction. Great. So hello, everyone, and welcome to this Centre for Personalised Medicine, or CPM, lecture. My name is Annika Lukeson. I'm the director of the CPM, and I'm Professor of Genomic Medicine at the University of Oxford. So before we get started and I introduce today's speaker, I just wanted to briefly introduce the CPM to anyone who's not familiar with it. Sorry, I can't see the list of attendees just now. We are, amongst other things, an outreach initiative based at the University of Oxford as a joint venture between the Wellcome Centre for Human Genetics and St Anne's College. Our aim is to think about various aspects of personalised medicine and then engage with students, scientists and the public about this. We host talks, events, we have a blog, and more recently we've created short vlogs and animations, so do have a look at our website at these recent editions. You can also find recordings of our previous talks, including Jennifer Doudna, Heidi Ream, Adam Rutherford and many others on our website. Please follow us on Twitter at CPM Oxford to find out about future events. And our annual lecture is coming up, given by Professor Andrew Hattersley in the Sheldonian Theatre on and online on Thursday, the 26th of May. So our speaker this lunchtime is Holly Eggington. Holly is a DPhil student in cancer sciences, studying the interaction between cellular compartments in colorectal cancer at the Wellcome Centre for Human Genetics. She's also a medical student at St Anne's College, and if that wasn't enough, this year Holly is president of the Oxford University Personalised Medicine Society. She is passionate about bridging the boundary between laboratory and clinical research, and in the future, she hopes to work as a clinician scientist in oncology. Today, the topic of our talk is plasticity in colorectal cancer. Take it away, Holly.
Great, thanks, Annika. Can you hear me okay? Brilliant, thank you. Okay, yeah, so I mean, that was a great introduction. I don't think she said anything I couldn't say, but as an introduction, yeah, I'm a medical student, so I've completed 4 out of 6 years of my medical training at Oxford. I'm currently intercalating on a DPhil in cancer science, which is funded by Cancer Research UK, and I'm doing that as I hope to in the future go into oncology and hopefully develop some new treatments to treat different types of cancer. I'm also this year currently president of the Oxford University Personalised Medicine Society. So we're a society who are affiliated with the Centre for Personalised Medicine, and we're very passionate about increasing student involvement in personalised medicine, in particular fostering interdisciplinary relationships between undergraduate and postgraduate students from different courses, so maths, computer science, and of course biology, chemistry, medicine. So just as a little introduction, I'm going to go right from the start and we're going to talk about what actually is cancer. So cancer is a disease in which some of the body's cells are growing uncontrollably and spread to other parts of the body. And cancer can theoretically occur in any tissue, although it's most commonly seen in tissues with high rates of stress or cellular turnover, such as the intestine, the lung, breast, skin and blood, among others. So you might hear people referring to a tumour. A tumour is often, it can be cancer or it can be a benign lesion, which means it's not causing disease. And many lifestyle factors do actually increase your risk of cancer. So the biggest one I'm sure everyone knows is smoking massively increases your risk of lung cancer, as well as head and neck, throat cancer and colorectal cancer. Obesity is highly associated with an increase in cancer incidence. Sun exposure is associated with increased risk of melanoma or skin cancer. And then some other factors associated with particular cancer types. So, for example, occupational asbestos exposure increases your risk of a type of cancer called mesothelioma, which affects the lining of the pleural cavity of the lungs. But cancer, while it's affected by many of these modifiable risk factors, it is predominantly a disease of age. As our population are living longer, we're seeing increased cancer incidence, and that's because people are more likely to get cancer as they become older. And this is particularly unfortunate as, unlike many of these other factors, age is not a modifiable risk factor. So why do we get cancer as we age? Well, cancer is a disease of our DNA. Our DNA is responsible for coding for proteins, and proteins are what make our cells function. So we're made up of lots of different types of proteins which have lots of different functions. And when we have mutations in our DNA, these proteins stop functioning correctly. They might start to make cells divide too much. They might stop cells dividing at all. And cancer happens when these mutations result in cells dividing too much, because as I said, cancer is when your cells are dividing uncontrollably. So when we have normal DNA, mRNA, which is what the type of RNA which codes for protein, produces the normal protein, and when it's mutated, we get these mutated proteins. Now, 1 or 2 mutations are fine, and our cells are very good at repairing mutations. We have a lot of DNA repair machinery. Problem is that as we age, as time passes, these mutations gradually accumulate in cells. So the first mutation might not result in disease, the second mutation might not, the third mutation might not. But as we get more and more of these mutations, eventually there's going to be a hit which pushes a cell from a mutated healthy cell to a cancer cell, which is now growing uncontrollably. And these mutations aren't random mutations. There's certain types of mutations that we know help cells survive. So the most common ones of these are we lose these things called tumour suppressors, which are responsible for stopping cells when they start dividing too much. There's also mutations that result in proliferative signalling, and this means that cells get signals telling them to divide more. There's mutations that mean that cells don't receive inhibition when they're actually, so when a cell divides too much, it undergoes something called senescence, which stops it dividing any further, and they can actually overcome this using something called replicative immortality. And these are something we call the hallmarks of cancer. So as cancers accumulate more and more of these hallmarks by mutations, they start to become more and more like what we consider a cancer. And how is cancer treated? Well, it's most commonly treated by surgery, chemotherapy, and radiotherapy. We're also developing lots of new types of treatments, so immunotherapy, which harnesses our immune system in order to kill the cancer. Specific targeted therapy, and this is particularly in the realm of personalised medicine. For blood cancers, you may have bone marrow transplantation. And there's also certain types of hormone therapy, which can actually harness receptors on the cancer to stop them growing. So colorectal cancer is a cancer of the intestinal tract. This is the large intestine and the rectum. Colorectal cancer occurs in the epithelium of these tissues. And what you see is tumour growth over time. So from an adenoma to a carcinoma. And this is illustrated here, where we see a change from our normal epithelium. A mutation's accumulated, in this case it's a mutation in a gene called APC, which results in formation of an early adenoma and a dysplastic crypt. And we'll talk a bit more about dysplasticity later. This is then accumulates further and further mutations, eventually developing into what we would consider a cancer. And a cancer is what is capable of spreading, invading surrounding tissue and causing disease. For colorectal cancer, it's actually increasing in incidence in the UK, and in particular among young people. And this is largely thought to be due to lifestyle factors. So colorectal cancer is known to have increased incidence in Western countries, and this is probably due to red meat consumption and increased rates of obesity. But each year we're getting 42,886 new cases of bowel cancer on average. And of these, around 16,000 people a year will die. Although 50% of people are surviving, it's very important to note that that means 50% of people are not surviving. And that's where we need to try and find new ways of treating colorectal cancer, which work out why these patients aren't surviving treatment. One of the interesting things of colorectal cancer and bowel cancer is actually we have a very good screening programme which is able to identify colorectal cancer. This is where people who are aged 58 and 57 and 60 plus in the UK will be sent a test which tests for blood in their faeces. And this means that cancer can be detected at an early stage. And this is really important because the earlier you detect a cancer, the better chance of survival. So this is because one of the predominant ways we treat colorectal cancer is surgery. So we actually remove the cancer. And if you remove a cancer before it started to develop, become a later stage cancer and spread, you have a much better chance of the cancer actually not relapsing, not remitting and not coming back. So if you can catch the cancer when it's an adenoma, which is just a small polyp, chances are you can actually remove the whole thing by surgery and you've cured the cancer. For later stage cancers, we often introduce other therapies, particularly chemotherapy and radiotherapy. And that's particularly if we think the cancer might have metastasized. Metastasis is when a cancer is spread to other organs in the body. However, use of molecular biomarkers to guide treatment for colorectal cancer is still relatively limited. So under NICE guidance, the only molecular typing of colorectal cancer which is actually recommended is testing for mutations in these genes called RAS and BRAF. And these are indicative of worse prognosis for anti-epithelial growth factor receptor therapy. This type of hormone therapy. And when we're using chemotherapy, the main thing we're targeting is the characteristic of cancers to divide. And this actually means that chemotherapy has a dose-limiting toxicity. If we give too high a dose of it, then our healthy cells, which need to divide, will also start to die. So if you think of chemotherapy and common side effects, you think of things like hair loss, and you think of things like intestinal damage. And there's a point past which this becomes unsustainable for the patient. So chemotherapy regimens under NICE guidance use these different regimens. One is an anti-metabolite. Oxaliplatin is then used alongside this to help prevent progression-free survival. And why does therapy fail? So, as I said, 16,000 people a year in the UK will die from bowel cancer. And that's in spite of the fact that they will have had whatever treatment is best suited for them. They'll have had chemotherapy, they'll have had surgery if suitable. And that's because cancers can be resistant to chemotherapy. So they can metabolize drugs, they can actually decrease drug influx, or they can increase drug efflux. Aspects of the tumour microenvironment may affect this. So tumours which are able to survive hypoxia, which is when tumours are resistant to a lack of oxygen, are much less likely to respond well to radiotherapy, which is dependent on development of these things called oxygen-free reactive oxygen species, which damage DNA. And part of the reason that therapy fails is because cancers consist of many different cell types, not just the cancer cells and not just one type of cancer cells. And more importantly, cancers evolve over time. So every cancer will be different. This means that if you're using a chemotherapy which targets 90% of the cells in a cancer, if just 10% of those cells survive, then it doesn't matter if you haven't cured the cancer can grow back. And additionally, factors from these things called fibroblasts and the extracellular matrix may actually increase the chance of a cancer surviving. We often don't characterize these when we're looking at genetic factors in a cancer, so you can actually miss factors which may make a cancer survive. And this is why personalised medicine is really important, because it means that we can theoretically in the future we'll be able to take biopsies of a cancer and take into account all of these different things, the different mutations in different parts of the tumour, factors of the microenvironment, and actually we can better fit treatment to a cancer. But as I said, chemotherapy has dose-limiting toxicity. So chances are, if we are doing this, it won't be to optimise chemotherapy regimens. It will be to optimise more personalised treatment. So we are working on better categorising colorectal cancer. So this really seminal paper by Eggington et al. Basically has introduced something called the consensus molecular subtyping, where we actually look at a colorectal cancer and we categorise it into one of these 4 subtypes based on mutations, based on microenvironmental factors, and these are associated with prognosis. So the CMS1 type and CMS4 type are both associated with worse survival. CMS1 with worse survival after relapse, and CMS4 with worse relapse-free and overall survival. And by harnessing this kind of subtyping, again, we can theoretically introduce more personalised treatment for cancer. So if we had a patient's cancer, perhaps we could identify that they had a CMS4 tumour, and we could say, okay, we think you're more likely to have worse overall survival, so we're going to introduce a more aggressive treatment right from the get-go. The problem is, again, heterogeneity kind of confounds this. So even these consensus molecular subtypes show a significant amount of heterogeneity between different regions of the cancer, between metastases and the primary tumour site, and over time. So we see evolution of tumours over time and between different sites. Again, this is partially going to be due to microenvironmental factors. A tumour that's in the liver is going to have a very different surrounding microenvironment than a tumour that's in the primary site of the intestine. It's due to the fact that tumours want to evolve. So one of the hallmarks of cancers I mentioned earlier is actually that tumours will select for an increased mutation rate. They will actually they will select mutations which damage their own DNA repair machinery in order to increase their mutation rate over time. And that's because the more mutations they have, the more likely they are to actually have some subclones, so single tumours or small number of tumour cells which have different mutations, which are likely to survive a selective pressure. So let's go back to this. Why does therapy fail? Well, one of the big issues we have in cancer is this thing called a stem cell, or a cancer stem cell specifically. So stem cells are cells which are capable of differentiating to a number of specialised stem cells, a number of specialised cell types, and more stem cells. So if we look at an embryo has stem cells which are capable of forming all of the cells in our body. As we grow and as we differentiate and as we become a full-formed human, our stem cells tend to lose that capacity. And instead we have stem cells that are specialised for the organ that they're in. So we have intestinal stem cells, brain stem cells, skin stem cells, and we also have cancer stem cells. So a cancer stem cell is a small subset of cancer cells that are responsible for tumour initiation and growth, possessing properties such as indefinite self-renewal, slow replication, intrinsic resistance to chemotherapy and radiotherapy, and an ability to give rise to differentiated progeny. So I think we can probably see how a cancer stem cell would provide a significant barrier to cancer treatment. It doesn't matter if you've attacked the whole cancer. If you've left the single cancer stem cell behind, then your tumour is able to relapse, because as I said, they're capable of forming all the other differentiated cell types of the tumour. Furthermore, they're resistant to chemotherapy and radiotherapy. So they're difficult to treat, and if you don't treat them, the entire tumour can come back. And these cancer stem cells are probably responsible for a significant number of deaths due to colorectal cancer. Cancers that have metastasized prior to notice, cancers that seemingly have been treated by surgery and adjuvant chemotherapy and then recur. So to understand the cancer stem cell in colorectal cancer, you first need to understand the normal stem cell. So the intestinal epithelium is actually a great tissue for studying cancer generally, because it's under the control of these really tightly regulated morphogen gradients. So morphogens are secreted signalling molecules, which are responsible for basically determining cell fate in the intestinal crypt. So in the intestine, we have these things called villi, which are responsible for absorption, and then the crypts. And the crypts at the base, we have the intestinal stem cell. As the morphogen gradients change, as we lose signalling of this molecule called Wnt and we gain signalling of this molecule called BMP, we see these stem cells at the base slowly differentiate into functional cells. They're maintained by another type of differentiated cell called pan of cells, which secrete other molecules, particularly Wnt, in order to maintain stem cell fate. And then we lose stem cells as we move up. So we only see stem cells in the crypt base under normal conditions in the intestinal epithelium. And stem cells in the intestinal epithelium, as I said, are capable of making all of the different functional cells. Absorptive cells, which are responsible for absorbing things like food and water, secretory cells, which are responsible for absorbing things like, so secreting things like mucus, which are needed for normal epithelial function. And the homeostatic crypt stem cell was identified by Barker et al. As the LGR5 crypt-based columnar stem cell. So demonstrated by this little guy who's resting here. These are the homeostatic stem cells, which are responsible for normal tissue turnover and maintenance when the tissue is at rest. However, recent work has actually identified there's a second functional stem cell population in the intestine. And we've dubbed this the regenerative stem cell population. And that's because these seem to appear in conditions of tissue damage. So if the tissue is damaged by some kind of disease, infection, physical damage, then we see these regenerative stem cells happen. And they're responsible for actually repairing the tissue once damage has happened. So cell plasticity is a really important thing to understand in the context of both inflammation and cancer. So cell plasticity is the ability of cells to change their phenotypes without genetic mutations, that's really important, in response to environmental cues. So plasticity endows cancer cells with the ability to shift dynamically between a differentiated state, which has limited tumorigenic potential, as I mentioned before, and a cancer stem cell-like state, which is actually responsible for maintaining long-term tumor growth. And it's actually coming across as a newly emerging hallmark of cancer. So the ability of a cancer to actually develop a plastic state is really important for actually helping a cancer to develop and adapt and respond to selective pressures. So let's go back to homeostasis and inflammation for a moment. So following tissue damage, plasticity is key for regeneration and restoration to homeostasis of tissue. So what this means is we have inflammation occur, and we see this rise in the regenerative stem cell population. Once the inflammation has been resolved, once the tissue has been repaired, we then see a restoration back to the crypt-based columnar cell population. But tumours have been described as wounds that do not heal. So pathways which are actually taking place in inflammation for healthy tissue repair are also being observed in cancers. Cancers are already known to be very inflammatory. They're full of stromal cells, immune cells, remodeling of the extracellular matrix, much like we see in normal tissue repair. And we know that chronic inflammation, so inflammatory bowel disease, is actually associated with a significantly increased risk of colorectal cancer. So chronic inflammation plus somatic mutations, so spontaneous mutations to DNA, significantly increase your risk of cancer. And we see a significant amount of disruption to all these different signaling pathways. So now we have this really interesting relationship. We have homeostasis, where we see these resting crypt-based columnar stem cells. We have regeneration, where we see these inflammatory stem cells, these regenerative stem cells. And then we have cancer, where we're kind of seeing a balance between the two. We're seeing both, and it's very dependent on all these other factors. It's dependent on the mutations, it's dependent on the extracellular factors, it's dependent on immune cells. And I think understanding how these pathways are being co-opted in cancer is really important for understanding how cancers are able to adapt. If we want to understand how a cancer is able to shift its phenotype over time and spatially and during metastasis, we need to understand how the microenvironment is actually affecting the balance between these stem cells. So stem cell types seem to play different roles in colorectal cancer progression. So the evidence seems to suggest that in the absence of your crypt-based columnar LGR5 cells, that we see tumour growth. So we see a loss of tumour growth over time. We don't see tumour regression, the tumour maintains, but what we do see is an inability of the tumour to actually increase in size and progress. Whereas when we see, whereas we are seeing an association of the regenerative stem cell with metastasis. So I've already mentioned the statistic before that 50% of people with colorectal cancer will develop some form of advanced disease. And it seems that the regenerative stem cell is playing a really key part in this progression. So plasticity of these cells is helping to drive metastasis. So what we know is that firstly, that either just a tumour that only has LGR5 cells or a tumour that only has regenerative stem cells doesn't seem able to maintain itself. We also know that metastases and primary tumour sites have both LGR5 and regenerative stem cells. So this immediately points the finger at plasticity. If we assume that the regenerative stem cell is the primary type of stem cell responsible for initiating metastasis, then the regenerative stem cell must be able to reform both the LGR5 and regenerative stem cell, as well as the differentiated cell populations at the site of metastasis. So plasticity poses a barrier to stem cell targeted treatments. So as I mentioned, we know that elimination of the LGR5 positive compartment of a tumour will stop cancer progression, but it doesn't cause regression. So what we see is a temporary halt to tumour growth, but without any actual loss of the tumour. Now, if this was a primary tumour, which could be removed from surgery, maybe that would be absolutely fine. The tumour would cease to grow. You could potentially delay surgery somewhat and then remove the primary tumour as before. The problem is if the tumour has already metastasised, this is going to be completely pointless because you won't know the site of metastasis and you won't be removing that by surgery. And we know that once the selective pressure is removed, the eliminated compartment is able to repopulate. So we've been seeing this in papers where actually once you remove LGR5 elimination, we see regression of LGR5 cells. They aren't eliminated forever. And this is exactly what we'll be seeing in tumours as well. And as we know, that once we have both LGR5 and regenerative stem cells, the tumour starts to grow again. It starts to increase its fitness. So we need to understand what kind of signalling pathways are affecting this ability of stem cells to move via plasticity mechanisms from one phenotype to the other. So morphogens, as I've mentioned before, are really important for the maintenance of cell fate in the healthy intestinal crypt. So we have the Wnt signal pathway, which is responsible for promoting stemness. And we know that actually Wnt overexpression, which is really commonly seen in colorectal cancer via a mutation in the gene called APC, which results in functional Wnt overexpression, results in cancer progression. So it acts as an oncogene. BMP acts as a differentiation factor. So as we move up the crypt, the cells differentiate, they lose their stemness, they become functional cells. And these non-stem cells are much less tumorigenic. So we know that mutations to BMP and BMP inhibitors result in certain cancer syndromes. So juvenile polyposis syndrome is associated with mutations in genes which affect the BMP signaling pathway. I'm particularly interested in the YAP signaling pathway. So YAP is extremely restricted to the crypt base, and it's known to be associated with stemness. So YAP-dependent reprogramming of LGR5 stem cells has been observed to drive intestinal regeneration in cancer. So what does that mean? It means that we think that YAP may be one of the key pathways. It's just responsible for this flux between the LGR5 stem cell and the regenerative stem cell. And we can see this in regeneration. If you look at tissue which is regenerating, which has been damaged, what we see is this increase in YAP from a crypt-based restricted expression to being expressed completely throughout the whole crypt. And we see this with a concurrent increase in regenerative stem cell markers. So a little bit more about the Yaptas signaling pathway. So YAP and Yaptas, they are both homologs of the same gene, and they are downstream of the Hippo signaling pathway. So the Hippo signaling pathway is a signaling pathway which is a developmental pathway, and it was originally observed due to its role in controlling organ size in animals through the regulation of cell proliferation apoptosis. And it was named so because you get tissue overgrowth in mutations in Hippo. So you get a hippopotamus-like phenotype is how it was named. And loss of Hippo pathway signaling is actually commonly observed across many cancer types. So it's viewed as a functional tumor suppressor, as is MST1 and 2, LATS1 and 2. They're all three mutated in cancers and have been given the role of a tumor suppressor. And this is likely due to the role of Yaptas downstream. So the Hippo signaling pathway and Hippo signaling cascade inhibits Yaptas signaling. Yaptas under homeostasis is actually trapped in the cytoplasm of a cell. So it's attached to tight junctions and adherence junctions, and it's sequestered in the cytoplasm, and it doesn't exert any transcriptional effects. Once you see a loss of Hippo signaling, you see this translocation of Yaptas to the nucleus, where it can have a really wide range of transcriptional effects. So Yaptas is associated with increased epithelial to mesenchymal transition, and this is a really key characteristic of metastasis. It's also associated with increased chemo resistance, again, going to pose a huge barrier to chemotherapy. It's also associated with this resistance to this process called anoikisis. I definitely said that wrong, which is a form of programmed cell death, which occurs in cells when they detach from the surrounding cellular matrix. So what this means is that as cells in an epithelium, which is a very tightly regulated tissue, detach from each other, normally they will die because they no longer have the cell-to-cell communication, which they are dependent on. However, when you have overexpression of Yaptas, cells don't respond to this. You lose the normal cell matrix interactions. And this is, again, really important for metastasis, because one of the key steps of metastasis is cells need to escape their epithelial state. They need to be able to actually leave the epithelium and spread in the bloodstream. So if Yaptas is promoting the regenerative stem cell phenotype, and it's also helping cells to survive metastasis, it seems very likely that the Yaptas pathway is really key in initiating metastasis. If it means that regenerative stem cells are able to spread and metastasize, then it follows that targeting the Yaptas pathway might prove a really effective way of actually preventing metastasis and treating metastasis. And what's particularly interesting about the Yaptas pathway is that it's actually also a mechanic transduction pathway. So not only is it integrating molecular signals from the Hippo pathway, it also has crosstalk with numerous other pathways. It also actually is able to respond to things like collagen in the extracellular matrix. And we know that extracellular matrix remodeling and microenvironmental modeling is a really key characteristic of all cancers, and we observe it particularly in colorectal cancer. So again, if Yaptas is acting as an integration hub of all these signals, it follows that it's a really promising target. But it also has some paradoxical roles, which we don't quite understand yet. So regenerative reprogramming of the intestinal stem cell state in this paper was actually observed to suppress metastatic colorectal cancer. They observed that when you have overexpression of YAP or higher expression of YAP, they were actually seeing a decrease in metastasis, which is very confusing. And you see this a lot in the literature of Yaptas, where some papers seem to show it having a pro-tumorigenic effect and some people seem to show it having an anti-tumorigenic effect. So it's very paradoxical and we can't quite make sense of it yet. But I think the key to understanding how it interacts with the pathways is actually going to be identifying what factors in the environment are affecting Yaptas function. So it's not so simple as saying that YAP overexpression increases cancer or YAP overexpression decreases cancer. It's YAP in the context of other pathways. So I'm particularly interested in the context of colorectal cancer of the interactions between YAP with the Wnt pathway and the TGF-beta pathway. And that's because these are the key pathways which we see in the intestinal epithelium. So we see Wnt and BMP is a member of the TGF-beta superfamily. And we know there's a lot of crosstalk between BMP and Wnt. They inhibit each other, which is how the Morphogen gradient is established. There's a lot of evidence to suggest that Wnt actually promotes Yaptas signaling, which would be very logical if Yaptas is indeed primarily oncogenic. There's also a lot of evidence for some crosstalk between BMP and Yaptas, but the exact nature of this isn't understood. And all we know is that somehow crosstalk between these pathways is ultimately acting to have tumorigenic effects. So we also need to understand that how these actual mechanical factors are acting on the Yaptas pathway. And we know that inflammatory cytokines actually can act to promote Yaptas signaling and in turn increase regenerative stem cells. So understanding all of this is kind of the focus of my research and actually working out how the Yaptas pathway might mediate cellular plasticity and colorectal cancer survival over time. Okay. Thank you.
Holly, thank you so much.